So children, good morning to all of you. What we did yesterday, it was about the amino acids, and I told you that there are 20 amino acids which are involved in protein synthesis. So in 12th class, if I there is a process of protein synthesis which is translation, and in that we say that there are 20 amino acids. It doesn't mean that in the body there are only 20 amino acids. There are 20 amino acids which are involved in the process of protein synthesis. So we have, I was telling you about the amino acids, which are monomers or units of proteins. So there are 20 amino acids, I'm writing AA, which are involved in the process of protein synthesis. 20 amino acids are involved in the process of protein synthesis, and this process of protein synthesis is translation. We will study this in class 12, translation. There are more amino acids in the body, and out of which about 100 amino acids are very much required by the body. 100 amino acids are required by the body. And of course, the few names of amino acids which are not involved in protein synthesis. The names I told you are citrulline, ornithine, etc. I'm just telling you two names, citrulline, ornithine. These are the two names of the amino acids which are not involved in the process of protein synthesis. Then I told you that there are some sulfur containing amino acids, and there are some amino acids in which hydroxyl group is present. So sulfur containing amino acids, very important. They have sulfur in them. So that is steam. This is the name of one amino acid. Another is methionine. Both these amino acids, they have sulfur in them. Then there are amino acids in which OH group is present. This OH group is present in amino acids, namely serine, threonine. So these are the amino acids in which OH group is present. Now, beta, yesterday I was about to start with the chemical structure of protein, of amino acids. So the chemical structure of amino acids. Amino acid has a basic group and an acidic group. Both basic and acidic group is present in amino acid. The basic group which is present, it is NH2, and the acidic group which is present is COOH. So two functional groups, one is basic, one is acidic, is present in the amino acid. And because of this, the structure which the amino acid has, it has a centrally located alpha carbon, then it has COOH attached, then it has NH2 attached. So we write it H2N. Then we have H here, and we have R here. So this is the chemical structure of amino acid. I think one word is not visible. Now it's clear to you. Now in this particular carbon is alpha carbon. This is variable group. I'm just putting arrow so that you understand this is the labeling variable group. This is the acidic group. This is the basic group. So this is the chemical structure of the amino acid. If we draw the expanded structure of this amino acid, it will appear somewhat like this. We have carbon in the center, we have this variable group, we have this hydrogen over here, and then we have one carbon which appears to be like this C double bond O OH. And here we have nitrogen which has two hydrogens along with this. So this is the expanded structure of amino acid. I think little is not visible to you. I'll take it apart. Just a minute, beta. I'm just trying to show you the expanded chemical structure of amino acid. Okay, I'll write again. I'll write somewhere else. I think this is not visible to you. I'll write over here. So what we have is carbon. Then we have the carboxylic group, which is in this form. We have the variable group, which is R. We have hydrogen. We have nitrogen, and this nitrogen are attached to hydrogens. So this is the expanded structure of amino acid. Is it clear to you, beta? Is it clear? Anything which you could not understand or is not clear to you? Yes, bache? Is it clear?
Yes, ma'am, clear.
Okay. Fine. So this is the expanded structure. Now, what do we mean by this variable group? This variable group can have different functional groups. Like if this R, I'll draw it up. If this R in some case is H, or this R is in another case, if this R is replaced by CH3, or this R is replaced by CH2OH, then what do we get? So if this R is replaced by H, like then the structure will be H2N C COOH H, and here R is replaced by H. If this is replaced by CH3, what we'll get? H2N C COOH H here, and here CH3. If this R is replaced by CH2H H2N C COOH H and CH2OH. So their names will change. If this is H, we call it glycine. If this is CH3, we call it, this amino acid is L-alanine. If this is CH2H, the amino acid is serine. So the variable group in case of glycine is H. The variable group in case of alanine is CH3. The variable group in case of serine is CH2OH. And this is the chemical structure of different the structure, chemical structure of amino acids with different functional groups. Now, the amino acids, I'll tell you the types. So for that, I'll erase this. The amino acids are of three types. They can be acidic amino acids. They can be basic amino acids, or they can be neutral amino acids. If the amino acid has two carboxylic groups, acidic amino acid presence of two COOH groups, then we call this acidic amino acid. That means the presence of two carboxylic group makes it acidic amino acid. So two carboxylic groups, two COH groups. So this will be a acidic amino acid. The example of this acidic amino acid is glutamic acid. The example is glutamic acid, or another example can be aspartic acid. When in an amino acid, two NH2 groups are present. When in an amino acid, two NH2 groups are present, such amino acids are known as basic amino acids. So, presence of two NH2 groups. Such are known as basic amino acids. The examples of basic amino acids are lysine, arginine. When in an amino acid, one amino group and one carboxylic group is present, such amino acids are known as neutral amino acids. So presence of one COH group and one NH2 group. These amino acids are neutral amino acids. The example of these amino acids are, valine is one such amino acid, cysteine is another such amino acid, and glycine is another such amino acid. So we have three types of amino acids, which depends on the presence of acidic or basic group in it. If two COH groups are present, then it is acidic, example glutamic and aspartic acid. If two NH2 groups are present, we call such amino acids basic amino acids, example lysine and arginine. If one COH group and one NH2 group is present, such amino acid is known as neutral amino acid, example valine and cysteine. So these are the three types of amino acids. This is how we distinguish the amino acids. So any query in this till now, beta? What I have taught, any query in this? Yes, bache? Yes, beta, any query, any question? Any question in this? Okay, Rohan? Amrita, Yashika? Yes, Amrita, Yashika? Yes, beta? Any other question? No. So fine. We proceed now. We proceed on to the next one, and that is the bonding. Bonding in bond formation in amino acids to form protein molecules. The bond which is formed between amino acids This is one amino acid, another amino acid, another amino acid, another amino acid. The bond which is formed like this between the amino acid, it depends, the length depends on what protein has been formed. In some protein the length is short, in some protein the length of this is long. So it depends which protein molecule has been formed. But when the amino acids are linked with each other by peptide bond, we say that a protein molecule has been formed. So this linkage between the amino acids, this is known as peptide bond. Now I'll tell you how these peptide bonds are formed in between the amino acid to form a protein molecule. So for that, I'll erase it. So we are on to the next topic, peptide bond. Peptide bond. How this peptide bond is formed. Now here, I told you two amino acids are linked together. Now, how this linkage takes place? Linkage is always COOH group of first amino acid is linked to the NH2 group of the second amino acid. I told you there is a amino group, there is another group, the COH group also. So the amino group is linked to the COH group. So now how this linkage takes place? The linkage is shown like this. This is H, another H molecule linked to N. Then we have C. Let's show it by the glycine. Let's take the simplest amino acid. Then C, double bond O, and here OH. This is one glycine molecule. So the glycine molecule, I'm showing it how it combines with another one, which is the L-alanine. So plus N, here H, here H, here C, CH3, H, C, double bond O, single bond OH. How this combine, how the peptide bond formation between the two takes place, this I'm going to tell you now. That means COOH of this will be linked to NH2 of this. This is the first amino acid, this is the second amino acid. The first amino acid is glycine and the second amino acid is L-alanine. So how these two are linked, this I'm going to show. So during the linkage, again, what we will get, I'm making it here, N, H, H, C, H, H, C, double bond O. During the linkage, this is this, I'm just, this and this participates. And what does it form? Only one H, it forms the bond, N, C, C, double bond O, OH, here H, here CH3, plus this OH and this, it forms one water molecule. So this is the peptide bond formation. Here the peptide bond formation has taken place with the release of a water molecule, with the release of a water molecule. Now here if you see the participation of the carboxylic group of first and the amino group of second, and this results in the formation of glycl-alanine. What is this formed is glycl-alanine. This is the peptide bond formation. Is it clear, children? Is it clear? Peptide bond formation. Okay, Priya. Okay, Rohan. Two responses only. Only two responses. Okay, Harsha. Many are there. Zainab, Lalit, Yashika, fine. Many students are there. Jasleen, Siksha, Mannat, Amrita, Sanvi, Ananya Thakur, Ananya Gupta is also there today. Lalit, Zainab. I want your response whether you are understanding or not. Is it clear, children? Okay, fine, Ananya. Okay, so this is the type of bond which is formed between the amino acid to form a protein molecule. So this is how. Now this will be linked to another. This from this portion only, from the CH, it can be linked to another amino acid, and then it can be linked to another amino acid. Thus, the long chain of amino acid, long chain of protein molecule is formed, which depends on which particular protein has been formed. Now, another property of the amino acid, which is very important and many a times it has been asked. So it is the Zwitter ion formation. This is mostly seen in the solution form, although sometimes rarely in the solid state, but mostly seen the amino acids in solution form. If they are dissolved form, amino acids. Of course, in solid forms also it is seen, but mostly in solution forms. This is most common. They form a Zwitter ion. The Zwitter ion. The amino acids exist in Zwitter ion form. Amino acids can exist in the Zwitter ion form, mostly in soluble state, but rarely in solid state. Now, what happens? See, the amino acids have both carboxylic, acidic carboxylic group and a basic amino group. I'll say amino acids, these amino acids, they have both. They have, amino acids have both acidic carboxylic group and basic NH2 group. Now, in the solution form or under certain conditions, what happens? There is an internal transfer. There is an internal transfer of H from COH to NH2 under certain conditions. There is an internal transfer of hydrogen. The hydrogen gets transferred from COH to NH2. The hydrogen from COH gets transferred to another NH2, and because of this, what happens? A ion is formed. Under certain condition, there is an internal transfer of hydrogen from COH to NH2, and this results in the formation of an ion. Results in the formation of an ionic form with both positive and negative charge. With both positive and negative charge, which is called as Zwitter ion. This ion formed is called as Zwitter ion. Now I'll tell you how does it look and what is a Zwitter ion. So normally, how does it look? I'll make a normal acid, this normal amino acid. So normal amino acid is H2N C COOH. Here H, I'm making here H glycine I'm making. This is a normal amino acid. This is a normal amino acid, the normal state of an amino acid, amino acid. In normal state, the amino acid appears to be like this. Now, in the zwitterion, in the soluble form, in the zwitterion, how does it look? So it becomes H3N with a positive charge, COO with a negative charge, H and H. So this has a positive charge and a negative charge. This is a zwitterion form. This is a zwitterion form of an amino acid. Can you see the difference between the two? The H from COH has got transferred to amino group. From carboxylic group, the hydrogen has got transferred to amino group. So what do we get? We get H3N with the positive charge and COO with the negative charge. This is a zwitterion form of an amino acid. Can you differentiate between the normal state of an amino acid and zwitterion form of an amino acid? Is it clear? Can you differentiate, beta? Yes, bache? Can you differentiate between the two? Okay, Priya. This is the zwitterion form. Yes, beta. Others? Yashika, Amrita, Harsha, Jasleen, Siksha. So many are there. Yes, bache. I'm not getting any response. Why? Come on, beta. Try to respond. Yes. Can you send the message again? Can you send the message again? Just now I got a message. I couldn't read it. Just send me the message again. Okay. Okay, Priya. Okay, Sanvi. Yashika, Harsha, Amrita. Okay. Now this is zwitterion. This is the zwitterion. Now, ma'am, what would you share the next practicals which are okay? I'll share. Yes, beta, I'll share the practicals very soon. Now, this is the zwitterion form. Now, we did a term, if you remember, and the term was hermaphrodite. In case of animal kingdom, we use this term hermaphrodite. You remember what is this hermaphrodite? Do you remember what is this hermaphrodite, beta? What was this term used for? Yes, what is this? What is this hermaphrodite? You remember we did this term in animal kingdom, male and female sex organs. Fine, male and female, but what about them? What about them? What about the male and female sex organs? In an organism, yes, now you are right. Both the sexes in an organism is hermaphrodite. So can you tell me, there are two more terms, dioecious, yes, Rohan, it's correct, earthworm, fine. Then two more terms, we did dioecious and monoecious. Which one can be dioecious and monoecious? Out of these two, which one can be used with hermaphrodite? Out of these two, which can be used with hermaphrodite, dioecious or monoecious? Monoecious, that's right, Amrita. When both the sexes are present in the same organism, we use the term hermaphrodite, or we also say monoecious. That is right. And the example is earthworm. Here, both the charges, if you see in the zwitterion, the positive and the negative charge, they are present in the same amino acid. So we call this a hermaphrodite species of amino acid. This is a hermaphrodite type or hermaphrodite species a, uh, of an amino acid. Now, because both the charges are present in same, and because of this, this is also electrically neutral. This is also electrically neutral. This is also electrically neutral. Now, what happens to the Zwitter ion under different pH? This I'm going to tell you. What happens to the Zwitter ion under different pH? Under different pH means when we add an alkali to it or when we add an acid to it, then what happens? So for this, I'll erase some of it. So I'm going to tell to the Zwitter ion what happens when an alkali is added. So when, first of all, I'm telling you the under different pH, what happens to the Zwitter ion? Under different hydrogen ion concentrations. So first I'm telling you when an alkali is added to it. That means we will be adding OH. We will be adding OH to it. What happens when OH is added to it?